Hello everyone, we are the 7th group from the BS Medical Laboratory Science 3-1 and today we are going to discuss the biological functions of complement proteins. Let me introduce first the people behind this discussion. First is Aliyah Audrey Abiera, Janelle Averion, Sahara Brookshield Centino, Crizel Lirio, Mignon Leonardo, Andrea Manalo, and yours truly, Michael Algozo.
What is a complement? Complement is a complex series of more than 30 proteins. Its primary function is to amplify inflammatory response to destroy and clear foreign antigens. These soluble and cell-bound proteins interact in a very specific way and have powerful abilities. In addition, complement has important housekeeping rules which recognize cellular debris such as apoptotic cells and immune complexes, tagging them for removal by innate immune cells. However, complement activation needs to be carefully regulated as chronic activation could lead to inflammation and tissue damage to the host. If deficiency occurs, complement system can result in the increased chance of infection and accumulation of immune complexes, which may result in possible autoimmune disorders.
With the complement proteins, these are those seen in the circulation in their inactive forms and mostly are synthesized in the liver. These complement proteins undergo three main physiological activities. First is to act in relevance to the host's defense against infections, which covers opsonization, chemotaxis, leukocyte activation, and lysis of bacterial cells. Second is to act in relevance to the interface between innate and adaptive immunity, wherein augmentation of antibodies and the enhancement of immunologic memory take place. And the last one, or the third one, is the disposal of wastes, where clearance of immune complexes from tissues and apoptotic cells is of being observed. For more in-depth discussions about this, we can also visit the videos of previous medical laboratory science groups. With what's mentioned early on, this table just shows selected complement components together with their biological functions such as the opsonization of phagocytosis in C3b and IC3b and lysis of cells in C5 to C9, and so on and so forth with C5a and the classic component pathways. At this juncture, let us proceed on the discussion of each main serum proteins of the complement system with their respective biological and immunologic functions.
To start off with our complement proteins and their biological functions, the complement activation can be divided into three main stages: the recognition unit, activation unit, and the membrane attack complex. Focusing first on the recognition unit, the first complement component to bind is C1. So this protein is a molecular complex of 740,000 Daltons, and it actually consists of three subunits, which are the C1q, C1r, and C1s, that are stabilized by calcium. C1q has a molecular weight of 410,000 Daltons and is composed of 6 strands that form 6 globular heads with a collagen-like tail portion. C1q recognizes the fragment crystallizable, or known as the Fc region, of 2 adjacent antibody molecules, and the globular heads of C1q must bound to it in order to initiate the classical pathway. Binding of 2 specific regions of C1q to IgG and IgM results for the C1q to be converted into its active enzymes. Autoactivation of C1r results from a conformational change that takes place as C1q is bound. So the binding occurs, which opens up the active site on C1r, which has a molecular weight of 85 kilodaltons. Once activated, C1r cleaves a thioester bond on C1s, resulting for the C1s to be activated. Activated C1r is extremely specific because its only known substrate is C1s. Likewise, C1s has a limited specificity, with its only substrates being C4 and C2. Once C1s is activated, the recognition stage ends.
In the classical pathway, the first stage that involved is C1, and once the C1 is fixed, the next components that undergo activation will be C4, C2, and C3, which are known as activation unit. The activation unit will start with C4, which is the second most abundant complement protein with a serum concentration of approximately 600 Ug per ml, and it consists of 3 polypeptide chains and has a molecular weight of 205. The C4 will be cleaved by the activated C1 molecule. C4 is cleaved by the activated C1s molecule in C1 into two proteolytic fragments which are known as C4a and C4b. C4b will then bind to the antigen-antibody complex and has a binding site for C2. After that, C2 is the next component to be activated. C2 is a single-chain glycoprotein with a molecular weight of 102. C2 also cleave into two fragments which is known as C2a, then it will bind to C4b. This bimolecular complex C4b2a is a C3 convertase of the classical pathway and serves to cleave C3 through the enzymatic activity in the C2a portion of the complex. The next components that will activate is the C3. C3 is the major constituent complement system and the plasma concentration is 1200 Ug per ml. It serves as the pivotal point for all the three pathways. The classical pathway C3 convertase. The C3 molecule is cleaved by the C3 convertase into C3a and C3b. The C3a fragment has potent biological activities. The C3b fragment concovalently attached to the C3 convertase, resulting in a trimolecular complex consisting of C4b2a3b. This multimolecular complex is called the C5 convertase of the classical pathway, and it is specific for cleaving C5.
For the serum protein C5, its function is to initiate the membrane attack complex. C5 consists of two polypeptide chains A and B linked by disulfide bonds. It is split off by C5 convertase into C5a and C5b. The C5b attaches to the cell membrane of the target cell to initiate the formation of the membrane attack complex. For the serum protein C6, it binds to C5b in the membrane attack complex. So once the C5b has attached on the target cell's cell membrane, the C6 attaches to the C5b in order to stabilize it. For the serum protein C7, what it does is it binds to C5b C6 in the membrane attack complex.
Other serum protein that belongs to the membrane attack complex are the C8 and C9. C8 is made up of three dissimilar chains joined by disulfide bonds and has a molecular weight of about 150,000. Its function is to start pore formation on the membrane. On the other hand, C9 is a single polypeptide chain with a molecular weight of 70,000. The carboxyl terminal end is hydrophobic, whereas the amino terminal end is hydrophilic. The hydrophobic part serves to anchor the MAC within the target membrane. The complex of C5b, C6, C7, C8, and C9 is known as C5b9 or MAC. If it is soluble, it is known as SC5b9. When formed, the MAC presents a pore of 70 to 100 angstroms that allows ions to pass in and out of the membrane. Destruction of target cells actually occurs through an influx of water and a corresponding loss of electrolytes. The presence of C9 greatly speeds this lysis. However, sufficient perturbation of the membrane can occur in the absence of C9 so that deficiencies in C9 appear largely benign.
Moving on to the alternative pathway, pathogens can be destroyed in the absence of antibody by means of the alternative pathway, which acts as part of innate or natural immunity. This pathway is important as an early defense against pathogens. Phylogenetically, this represents the oldest of the C3 activating pathways. It was first described by Pilomer and his associates in the early 1950s. The pathway was originally named for the protein properdin, a constituent of normal serum. Now, it is known that properdin does not initiate this pathway, but rather stabilizes the C3 convertase formed from activation of other factors. The alternative pathway shows points of similarity with the classic sequence. Both pathways generate a C3 convertase that activates C3 to provide the pivotal event in the final common pathway of both systems. However, in contrast to the classic pathway,
The alternate complement pathway is predominantly a non-antibody initiated pathway. Triggering substances of the alternative pathway include bacterial cell walls, especially those containing lipopolysaccharide, fungal cell walls, yeast, viruses, virally infected cells, tumor cell lines, and some parasites, especially trypanosomes. All of these can serve as sites for binding the complex C3-BBB, one of the end products of this pathway. The three major components of alternative pathway are factor B, factor D, and proper D. Factor B contains a serine protease domain, and when activated, it provides the catalytic activity of the alternative pathway C3 and C5 convertases. Factor B circulates as an inactive proenzyme and only becomes activated after cleaved by the protein factor D. So the main goal of factor B is to bind to C3b to form C3 convertase. On the other hand, factor D circulates in the plasma as a mature but resting state form at a very low concentration and is produced mainly in adipocytes. The function of factor D is to cleave its unique substrate, which is the factor B, to generate the alternative pathway C3 convertases, C3H2OBP, and C3BBB. Lastly is the proper D, which stabilizes the C3 convertases, C3BBB. It is a key positive regulator of the alternative pathway by significantly increasing the half-life of the C3 and C5 convertases. It also plays a role in the efficient uptake of immune complexes. So to summarize the alternative pathway, C3 is hydrolyzed by water to produce a C3b, sometimes called IC3. This molecule can bind factor B, and when factor B is bound to C3b, B is cleaved to form C3BB, an enzyme with C3 convertase activity. More C3 is cleaved to form more C3BBB. This enzyme is stabilized by proper D, and it continues to cleave additional C3. If a molecule of C3 remains attached to the C3BBB enzyme, the convertase now has the capability to cleave C5. The C5 convertase thus consists of C3BBB, 3BP. After C5 is cleaved, the pathway is exactly the same as the classical pathway.
Let's now proceed with the MBL pathway. The MBL, which stands for mannose binding lectin or the lectin pathway, is one of the three pathways by which the complement system can be activated. This pathway is initiated by the binding of mannose binding lectin, colectin 11, and ficolins to microbial surface, oligosaccharides, and acetylated residues, respectively. The lectin pathway is activated by carbohydrates present in microbial cell walls and serves as an important link between the innate and adaptive immune responses. Molecules distinct to the lectin pathway includes mannose binding lectin or the MBL, MASP1, and MASP2. It acts as opsonine through binding to the collection receptor and phagocytes, and also activates the complement system, leading to the direct killing of pathogens. First up is the MBL or the mannose binding lectin. It has a molecular weight of 200 to 600 kD and a concentration of 0.0002 to 10 Ug per ml. Its function is to bind to mannose and initiate the lectin pathway. MBL binds to terminal mannose residues of microbial carbohydrates. MBL binds to a range of sugar including acetyl-D-glucosamine, mannose, N-acetylmanosamine, fucose, and glucose. This permits the protein to interact with a wide selection of viruses, bacteria, yeast, fungi, and protozoa decorated with sugars. An MBL-associated protease activates C4 and C2 as in the classical pathway. Next is the MASP1 and 2. MASP stands for MBL-associated serine protease. MASP and the MBL or lectin pathway are proteins that pair with homodimers or MBL-associated serine proteases. MASP1 and MASP2, which is analogous to C1R and C1S in a calcium ion-dependent complex. The majority of MBL in plasma is associated with either MASP1 or MASP2. The vehiculins associate with the MASP proteins to activate the lectin pathway. MASP1 or the MBL-associated serine protease 1 has a molecular weight of 93 kD and a concentration of 1.5 to 12 mg per ml. MASP1 is a serous protease that functions as a component of the MBL or the lectin pathway of complement activation. MASP1 is synthesized as a zymogen and is activated when it complexes with the pathogen recognition molecules of lectin pathway, the mannose binding lectin, and the vehiculins. MASP2 or the MBL-associated serine protease 2 has a molecular weight of 76 kD and has an unknown concentration. It cleaves C4 and C2 forming the C3 convertase and has been considered as the autonomous initiator or the proteolytic cascade, and it is associated with the pneumococcal diseases.
And now we will head to the receptors, and the following that will be mentioned are found on host cells that amplify and enhances immune response by augmenting phagocytosis and stimulating accessory cells rather than acting as regulators.
CR1, which stands for complement receptor type 1 or also known as CD35, is a large polymorphic glycoprotein which has a molecular weight between 165,000 and 280,000. It is found mainly on peripheral blood cells including neutrophils, monocytes, macrophages, erythrocytes, eosinophils, B lymphocytes, some T lymphocytes, and follicular dendritic cells. So it binds C3b and C4b but has the greatest affinity for C3b. Once bound to CR1, both C4b and C3b can then be degraded by factor 1. Its main functions are acting as a receptor towards red blood cells and platelet, and to mediate transport of C3b-coated immune complexes to the spleen and liver. It is there that fixed tissue macrophages strip the immune complexes from the red blood cells, process the complexes, and return the red blood cells intact to circulation. The ability of cells to bind complement-coated particles is referred to as immune adherence.
The ligands for CR2 include degradation of C3b such as C3dG, C3d, and IC3b. The cell types of CR2 are B cells, follicular dendritic cells, and epithelial cells. The function of CR2 is B cell receptor for antigen with CD19. Acting in concert with CD19, it binds complement-coated antigen and cross-links it to membrane immunoglobulin to activate B cells. With this manner, immune complexes are more effective at enhancing B cell differentiation and production of memory cells than is antigen by itself. CR2 plays an important role as a part of the B cell coreceptor for antigen. Also, CR2 is present only on mature B cells and it is lost when the conversion to plasma cells occurs.
For the complement receptor 3 or CR3, its ligands are IC3b, C3d, and C3b. CR3 is a receptor found on monocytes, macrophages, neutrophils, and killer cells. It plays a key role in the mediation of phagocytosis of particles coated with complement fragments. It triggers surface adhesion and increases the activity of phagocytic cells. The lack of these receptors results to failure in exhibiting chemotaxis, surface adherence, aggregation, as well as phagocytosis.
The complement receptor type 4 or CR4 is very similar to the complement receptor type 3. Both binds to the IC3b fragments in a calcium-dependent fashion. CR4 proteins are found on neutrophils, monocytes, tissue macrophages, activated T cells, dendritic cells, NK cells, and activated B cells. Neutrophils and monocytes, however, possesses smaller amounts of CR4 than of CR3. Their function appears to be similar to that of CR3, and they may assist neutrophil adhesion to the endothelium during inflammation.
Now, the last receptor, namely the MCP-CD46, has the ligand of C3b and C4b. It mainly serves as an inhibitory complement receptor and also has a cofactor activity through factor 1 cleavage or inactivation of its ligands, which is C3b and C4b. For its cell types, it can either be neutrophils, monocytes, macrophages, platelets, T cells, B cells, and even endothelial cells. And in practical sense, it has been involved in the clinical investigations of malignant brain tumors, especially in children, and prostate complications in adults.
For the overall summary of the topic, complement pertains to a series of serum proteins that are presented normally in our system and having the main function as the mediator of inflammation. Along with it, the functions of complement also include opsonization, chemotaxis, and lysis of cells. So there are 9 mentioned proteins from the classical pathway wherein they're activated by bound antibodies in a sequence as they are triggered by antigen-antibody combination. The following are grouped based on its certain reactants, dividing them as the recognition unit, activation unit, and the membrane attack complex. An additional number of serum proteins are involved in the alternate pathway, or also known as the Properdin system, that is triggered by the presence of microorganisms. These serum proteins help in stabilizing a key enzyme complex formed along the pathway. Lastly, the lectin pathway, which also serves as another antibody-independent system of activating complex proteins through its constituent mannose-binding lectin, or the MBL, is adhering the mannose that are found primarily through the outer coating of microorganisms. An additional information towards the complement receptors, which are the CR1, CR2, CR3, and CR4, is that these amplify and enhances immune response by augmenting phagocytosis and stimulating accessory cells rather than acting as regulators.
Now, these are the references that we have used in this discussion, and that is all of our discussion. We hope you have learned something from this video. And again, for background and in-depth analysis of pathways and acute phase proteins, please visit the videos that has been done by other groups within our class. Again, thank you for listening and keep safe.